Good evening, everyone. I'm Shivangi Patel, member of Shivi Forensics. Today, we will discuss MCQs on restriction fragment length polymorphism. So, before discussing MCQs, first of all, we will discuss what exactly is restriction fragment length polymorphism is. So, restriction fragment length polymorphism, commonly known as RFLP. RFLP is one of the earliest DNA-based molecular techniques used to detect genetic variations between individuals. It works by cutting DNA with restriction enzymes, that is, restriction endonuclease enzymes, and analyzing the resulting DNA fragments length. Differences in this fragments pattern arise due to genetic mutations, making RFLP a valuable tool in DNA fingerprinting, paternity testing, genetic disease diagnosis, gene mapping, and early forensic investigations. Although it has largely been replaced by PCR-based methods, RFLP remains an important milestone in the development of molecular genetics and forensic DNA analysis. Now, we will discuss one by one MCQs. So, the first question is restriction fragment length polymorphism is primarily based on RFLP is based on variations in the length of DNA fragments produced after digestion with restriction enzymes, and that is restriction endonuclease enzymes. Genetic mutations can create or remove restriction sites, resulting in DNA fragments of different sizes. And these fragment length differences are then analyzed to identify genetic polymorphism. So, the correct answer is variations in DNA fragment length after restriction enzyme digestions. Now the second question is, which enzyme is essential for performing RFLP analysis? So restriction endonuclease are the enzymes that recognize specific DNA sequences known as recognition sites and cut the DNA at or near these sites. In RFLP analysis, these enzymes generate DNA fragments of different length depending on the presence or absence of the restriction sites. And these fragments length differences are then separated by gel electrophoresis and analyzed to detect genetic variations. So the correct answer is restriction endonuclease. Moving on to the third question. RFLP polymorphisms arises mainly due to RFLP polymorphism mainly occurs because of genetic mutations such as single nucleotide changes or small insertions or deletions that alter the DNA sequence recognized by restriction enzymes. And if a mutation creates a new restriction sites, the DNA is cut into smaller fragments. If it is removes an existing restriction sites, larger fragments are produced. These differences in fragment length are the basis of RFLP analysis and are used to identify genetic variations among individuals. So the correct answer is mutations that create or abolish restriction sites. Now, fourth question is, which technique is traditionally used after electrophoresis in RFLP analysis? After gel electrophoresis, the separated DNA fragments are transferred onto a membrane using Southern blotting. A labeled DNA probe is then hybridized to a membrane to detect the specific DNA fragments of interest. So the answer is Southern blotting. Now moving on to the fifth question. The separation of restriction fragments in RFLP is achieved by, in RFLP analysis, restriction DNA fragments are separated according to their size using agarose gel electrophoresis. Smaller fragments migrate faster and farther than the larger fragments, allowing the DNA fragments pattern to be analyzed. So the answer is gel electrophoresis. Moving on to the sixth question. Which scientist developed the RFLP techniques? David Botstein. In 1980, David Botstein and his colleagues introduced RFLP as a genetic marker for detecting DNA polymorphisms. And this technique became an important tool for gene mapping, genetic studies, and early DNA fingerprinting. So the answer is D, that is David Botstein. Now, seventh question. Which type of DNA is generally required for successful RFLP analysis? RFLP requires intact non-DNA degraded DNA because restriction enzymes need complete recognition sites to cut the DNA accurately. Degraded DNA produces incomplete fragments, making the result unreliable. So the answer is high quality, high molecular weight DNA. Now, moving on to the eighth question, which of the following is not an application of the RFLP? RFLP is a DNA-based technique used for DNA fingerprinting, paternity testing, and gene mapping. It analyzes variations in DNA fragments, whereas protein sequencing determines the amino acid sequence of proteins and is not an application of the RFLP. So the answer is protein sequencing. Now moving on to the ninth question. Which of the following is a major limitations of the RFLP? A major limitation of RFLP is that it requires a large amount of high quality intact DNA. If the DNA is degraded, restriction enzymes cannot cut it properly, resulting in inaccurate and the unreliable fragments pattern. So the answer is requires large quantities of non-degraded DNA. The last question, which is 10th one, which sequence of steps correctly represents the RFLP workflow? The RFLP workflow begins with DNA extractions followed by the restriction enzyme stations. The resulting DNA fragments are separated by gel electrophoresis, transferred to a membrane using southern blotting, hybridized with a labeled DNA probe, and finally detected to analyze fragments length polymorphism. So the answer is B, which is DNA extractions, then restrictions and digestions, gel electrophoresis, southern blotting, probe hybridization, and at last detections. Thank you all of you.